Right now, as we speak, there's a massive scientific search party happening, and it's deep inside our own DNA. Scientists are literally hunting for these tiny, tiny clues that could unlock some of the biggest mysteries of our health.
So, let me ask you this. What if just one single typo in the 3 billion letter code that makes you you? What if that one little mistake could tip the scales on your risk for major diseases? For decades, this wasn't just some hypothetical question. It was an absolutely monumental challenge for geneticists. And to really get a grip on how huge this challenge was, geneticist Dr. Ryan Toohey described it perfectly. He said, it's like searching for a single typo on just one page of a massive book. That's the kind of scale we're talking about. It's what scientists were up against trying to find the exact genetic culprits behind all these complex diseases. All right, so let's officially start our detective story. First up, we've got to understand the crime scene, this mystery of genetic typos and, how we've been trying to find them for years. For almost 20 years, the main tool in the toolbox was something called genome-wide association studies, or GWAS for short. Just think of them as these giant genetic surveys. And they were super successful. They could scan thousands of people's DNA and flag entire neighborhoods in our genetic code that seemed to be linked to diseases like type 2 diabetes or heart disease. But, and this is the crucial problem, GWAS could tell us the neighborhood where a potential suspect was hanging out, but it couldn't give us their exact street address. We're talking about regions that could stretch for tens of thousands of DNA letters, and any one of them could be the culprit. So we knew the general area, but we had no way of identifying that specific single typo, the causal variant, that was actually making all the trouble. And that's where things were stuck for a long time. Until, that is, a new hero entered the story. A totally innovative technology that acts like a genomic speed reader. And it's what finally allowed scientists to crack the case. I mean, the leap forward here is it's incredible. The old way of checking out these genetic suspects was so painstakingly slow. You had to test them one by one. The new way? Well, it lets researchers test over 220,000 of them all at once. It is a complete and total game changer, both in scale and in speed.
Okay, so how does the speed reader even work? It's got a fancy name, Massively Parallel Reporter Assay, or MPRA, but let's break it down. Step 1, scientists grab thousands of these potential DNA typos. Step 2, they stick a unique molecular barcode, like a little price tag, onto each and every one. And finally, step 3, they put them all into cells just to see what happens. By scanning the barcodes, they can see exactly which typos crank a gene's activity up and which ones turn it down. It's really the ultimate way to see if a tiny typo actually changes the meaning of a whole genetic sentence.
So, after running this absolutely massive experiment, what did the code reveal?
Yeah, let's get to the good part, the results.
First off, the sheer scale of this investigation is just mind-blowing. The team didn't just look at a few dozen suspects. No, they put nearly 250,000 potential genetic variants under the microscope. And they didn't just do it in one setting. They tested them across different kinds of cells, like brain cells, liver cells, and blood cells. And from that massive lineup, they got over 13,000 confirmed hits. Wow. These are the typos that were proven without a doubt to actually change how a gene behaves. Think about it. This one experiment managed to solve about 20% of the mystery regions they were looking at, finally pinpointing the specific changes that really matter.
Now, the investigation totally could have ended right there. I mean, that's a huge success. But the data revealed something unexpected, a real plot twist that changed how we think about the way genetics works.
So it turns out, it's not always just one bad guy acting alone. In about 11% of the cases, the team found that some of these variants actually work together, like a team. The combined effect of two nearby typos was way bigger than what you'd get by just adding their individual effects together. It's like some genetic risks are less of a single-person job and more of a conspiracy.
Okay, this is the point where the story really shifts from just a fascinating scientific mystery to something with real-world impact. The big question is, how do we take all these amazing genetic clues and actually turn them into potential cures?
And this is exactly why it matters so much. The researchers were able to draw a direct line from their findings to our health. For instance, they found pairs of variants that, when they work together, actually lead to lower levels of bad cholesterol. They found other combinations that mess with a gene linked to blood pressure. And they even saw the same team-up effect with variants near a gene that's been associated with developmental disorders. And the impact just keeps getting broader. Get this. In one case, they zeroed in on a variant for blood sugar control that was first found in people of European ancestry, but because they understood how it worked, they could predict that similar variants found in people of African ancestry would probably do the same thing. And guess what? They were right. It just goes to show how absolutely vital it is that we study genetics across all ancestries so that these breakthroughs can benefit everyone.
So what does this all mean for the future? Well, this study isn't just the end of a mystery, it's really the beginning of a whole new chapter in medicine.
Dr. Lila Siraj, the lead author on the study, she said it perfectly. For years, all we could see was a statistical link, an association between some genetic neighborhood and a disease, but we didn't know why. This work, she says, is the bridge from that simple association to the actual biology. It finally explains how that link really works.
And here is where things get really futuristic. This massive dataset, with all 13,000 plus of those functional variants, is now being used as a training manual for artificial intelligence. The whole idea is to teach an AI how to predict the function of variants we haven't even had a chance to test yet. It could accelerate discovery in ways we can barely imagine. And this all leads us to the ultimate goal here: precision medicine. I mean, if you understand exactly which typo affects which gene in which type of cell, you can start to imagine a future where medicines are designed with surgical precision. They could be engineered to act only in the specific tissues where they're needed most. That means way more effective treatments and way fewer side effects. So this incredible research has basically given us the ability to read our own genetic code with a clarity we've never had before. And it leaves us with one final, very powerful question. Now that we have this amazing new roadmap, how soon can we go from just reading these typos to actually correcting them?